Good morning to all. I am Dr. M. Ainul Rifaya, working in Erode Sengundar Engineering College. Today we are going to discuss about the topic SAGE. SAGE is the serial analysis of gene expression. Okay, and then first in SAGE we are discuss about the fundamental of bioinformatics. So, what is bioinformatics? Bio represents the biology, informatics is the IT, and then bioinformatics is the interdisciplinary field. It includes biology, microbiology, biochemistry, genetics, molecular biology, and then computer science also. So it is called interdisciplinary field. It include all information related to biological information. Okay. So what are the applications in bioinformatics? First application is the main one, drug designing. Drug designing is the main goal of bioinformatics work. And the number of applications are available. Okay. Then our topic is SAGE. It is a serial analysis of gene expression. In SAGE, already I told what is mean by SAGE. So, what is SAGE? That is a serial analysis of gene expression is one of the powerful tool. This tool allows digital analysis of overall gene expression patterns and then produce a snapshot of the mRNA population in the sample of interest. So, particularly, SAGE provides quantitative and comprehensive expression profiling in a given cell population. Okay, so SAGE invented in the year 1995. SAGE invented in the year 1995. Invented at Johns Hopkins University in USA by Dr. Victor. And the main overview of this work, cells complete gene activity. They will provide the addresses specific issues such as determination of normal gene structure and identification of the abnormal genome changes. So here mainly we are focusing the comparative studies. So normal gene expression எப்படி இருக்கும் and then அப்புறம் abnormal ஜீனோட ஸ்ட்ரக்சர்ஸ் எப்படி இருக்கும் அப்படிங்கிறத தான் ஃபஸ்ட்டு நம்மளுக்கு தெரியும். It enables precise annotation of existing genes and discovery of new genes. And the main one is gene expression refers to the study of how specific genes are transcribed at a given point in time in a given cell. And then examining the transcripts are present in a cell. So SAGE is the main thing. It's a large scale studies of DNA expression. This can be used to create a expression profiles. So it is allow the rapid detailed analysis of thousands of transcripts of in a cell and then comparing a different type of cells. Generate profiles that will help to understand healthy cells. ஸோ ஹெல்த்தி செல்ஸு எப்படி ராங் அண்ட் தென் எப்படி இன்ஃபெக்ட் ஆகுது, டிசீஸ் டைமிங்கில் எந்த அளவுக்கு அதில் சேஞ்சஸ் நடக்குது அப்படிங்கிறத கம்ப்ளீட்டாக படிக்கிறதுக்கு தான் இந்த SAGE டெக்னிக் நம்ம யூஸ் பண்ணுறோம். ஸோ அண்ட் தென் SAGE மெத்தடாலஜி. ஸோ மெயின் ஒன் இஸ் த ஷார்ட் சீக்வன்சஸ் டேக். அதோட லென்த் பார்த்திங்கன்னா 10 to 14 பேஸ் பேர்ஸ். The base pairs contains a sufficient information to uniquely identify a transcript provided that the tag is obtained from a unique position within each transcript. And the main thing, sequence tags can be linked together to form long serial molecule that can be cloned and sequenced. And then quantitation of the number of time a particular tag is observed provides the expression level of the corresponding transcript. And then here see this diagram. First one is the sequences. Here we are isolate the SAGE tags. First step is the isolate SAGE tags. So isolate பண்ணிட்டு and then next step is link tag together. ஸோ அந்த லிங்க் பண்ண டேக்கை என்ன செய்கிறோம், சீக்வன்ஸ் லிங்க்டு டேக்ஸாக நம்ம இது பண்ணுறோம். ஸோ திஸ் ரிசல்ட் is a quantitative tags and determination patterns of gene expression. See this diagram. And then normal gene product and then abnormal gene product and then infected also. ஸோ இதுக்கு இருக்க கம்பேரிட்டிவ் ஸ்டடீஸ் நம்ம தெரிஞ்சுக்கலாம், எந்த ஜீனில் என்ன சேஞ்சஸ் நடந்திருக்கு அப்படிங்கிறத இது மூலயமா நம்ம தெரிஞ்சுக்கலாம். ஓகே, ஸோ வாட் ஆர் த டெக்னிக்ஸ்? எக்ஸ்டென்சிவ் சீக்வன்சிங் டெக்னிக்ஸ் இருக்குது வேணும் நமக்கு. அதே மாதிரி பயோ இன்ஃபர்மேட்டிக்கை பற்றின பேசிக் நாலேஜும் இருக்கணும். ஸோ தட் இஸ் அ பவர்ஃபுல் கம்ப்யூட்டர் சாஃப்ட்வேர். Assemble and analyze result from SAGE experiments in steps. And then first one is the isolate the mRNA of an input sample. For example, we are collecting the tumor sample. And then second step, extract a small chunk of sequences from a different position of each mRNA molecule. And then next step is the link this small pieces of sequences together to form a long chain, or it is called concatenamer. So and then clone these chains into a vector which can be taken by a bacteria. We are isolating the vector from the bacteria. Sequence this chain is using modern high throughput DNA sequences. And then here process this data with a computer to count the small sequence tags. And then see this diagram. Particularly, so this RNA is extracted from the cell. Here next step is the cDNA synthesis. Immobilized to streptovidin beads will included. And then next step is the restriction with anchoring enzyme. And then provide the part. And then this part we can like it to using the linkers use பண்றோம். And then next step, restriction with tagging enzymes and blunt ends. So and then next step is the ligation procedure, provide the result. And then next one is the produce the multiple copies using the PCR. What does mean by PCR? PCR represents the polymerase chain reaction. Okay, so and then PCR amplification, restriction with anchoring enzyme again we can produce the product. And then concatenation, linking strictly ends and clone. SAGE techniques. Trap RNAs with beads. That is one of the technique. And then first point is mRNA. It represents the messenger RNA end with a long string of AS. And then it will be adenine forms very strong chemical bonds with another nucleotide, thiamine. And then molecules that contains the 20 or of 20 or so TS acts like a chemical bait to capture RNAs. And then researchers coat microscopic magnetic beads with the chemical baits. When the contents of cells are washed, here next step is the content of the cells are washed past the beads, the RNA molecule will be trapped and the magnet is used to withdraw the beads and the RNAs out of the soap. And then here see this diagram. Tag that binds to mRNA and then mRNA also there. And then next step is a cDNA synthesis, complementary strand. So, double stranded cDNA is synthesized from the extracted mRNA by means of biotinylated oligoprimers. And then cDNA synthesized is immobilized to streptavidin beads. Here, see this DNA molecule and then mRNA will be present, enzymatic cleavage of cDNA. So, the cDNA molecule is cleaved with a restriction enzyme. And the next step is that we are using the type 2 restriction enzyme. So, also it is known as anchoring enzymes. Okay, and then 4 base recognizing enzyme used here. And then average length of cDNA 256 base pairs with sticky ends created. And then see this diagram. The enzymes play a role molecular scissor. The enzyme playing a role molecular scissor and enzyme cuts the DNA. Particularly the molecular scissor cuts the DNA. So, and the ligation of linkers to wait a minute. Ligation of linkers to bound cDNA, and these captured cDNAs are divided into 2 halves, then ligated to linkers A and linkers B respectively at their ends. Linkers also known as the docking molecule. They are oligonucleotide duplexes. And then linkers contain the nucleotide cohesive overhang. And then type 2s recognition sequence. And then third one is the PCR primer sequences. And then divide in COF ligate to the linkers. Here A and B. Divide the half ligate to linkers. First one is the A, second one is the B, and then type 2s restriction enzyme, that is a tagging restriction enzyme. It's called type 2s restriction enzyme is otherwise called tagging enzyme. And then here linker or docking molecules providing the result tag, and then enzyme. Tagging enzyme usually cleave the DNA 14 to 15 nucleotides. It will be releasing the linker adopted sage tag from each cDNA. and then repair of ends to make ended tags using DNA polymerase and TNTPS. See this diagram. An enzymatic locks onto the DNA and cuts off a short lock, and then DNA with the help of enzyme, that is a molecular scissor. We are here the docking molecules also present, and then formation of tie tags. So next step is the formation of tie tags. Its left is a collection of short tag taken from each molecules. Two groups of cDNAs are ligated to each other to create a tie tag with linkers on either end. See this diagram. Restriction with tagging enzymes, and then this enzymes providing the parts, and then tail to tail ligation. Tail to tail ligation. So, and the ligation using T4DNA ligase. See this diagram. Two tags are linked together. See this diagram. Two tags are linked together, and then tie tags. The linker tie tag linker constructs or amplified by PCR using primers specific to the linkers. See this diagram. Cleave with tagging enzymes and the blunt end, it will be produce the result ligate and amplify with the primers A and B. So why we are using in PCR polymerase chain reaction, we are providing a number of copies. And then isolation of tie tags. The cDNA is again digested by the AE, breaking the linker of right whether it was added in the beginning. See this enzyme, the cutting enzymes playing a role, molecular scissor, the docking molecules are removed, and then DNA will be present. So on the last எண்டில் இருக்கிற டாக்கிங் மாலிகுலர் நம்ம மாலிகுலர் சிசர் அதாவது என்சைம் யூஸ் பண்ணி கட் பண்ணிடுறோம். ஓகே, ஸோ cloning, concording and sequencing. Lots of copies are required, so the concordimers are put into bacteria. It will be act like living copy machines to create millions of copies from the original, and these copies are then sequenced. So these copies are sequenced using the machines that can read the molecules in DNA, and the result is a long list of nucleotide that has to be analyzed by the computer, and the analysis will do a several things. Count the tag, determine which one comes from the same RNA molecule, and figure out which one comes from known well studied genes and which one are new. See this diagram. That is a concordimerase tag into a plasmid for automated sequencing technology. So, and then extract and count tags to calculate the expression level of each transcript, and with the help of the softwares we to compare a tag counts to find differentially expression genes and identify full length transcripts from the sequence databases. Everybody know about the what is databases. That is a collection of data sets. There are two types of databases, primary databases and the secondary databases. Okay, so primary databases represents the original data, and then secondary databases it will be modifications. Okay, so original data வச்சு we are using information, so modification and then changing the data, and then quantitation of gene expression and data presentation. See this result, sequence analysis, and then normal one and then tumor one. So x-axis is the normal and then y-axis is the number of tags. We are finding the result. See this diagram. This is a simple model of the SAGE techniques. And then first step is the isolate of mRNA. mRNA represents messenger RNA. And then next step we are adding biotin labeled TT primer. And then this primer synthesis of TSCDNA and the bind to streptovidin coated beads and the cleave with anchoring enzyme. So and then unwanted fragments we are removing that, discard loose fragments and then divided into two pools and add linker sequences. See this diagram, here we are using the linker, two pools and add linker sequences and then ligate format and the cleave with tacking enzyme. See this, with tacking enzyme, combine pools and ligate. And then next one is the amplified Titacs, then cleave with anchoring enzymes, enzymes. And then next step is the ligate the Titacs and sequence and record the tags or frequencies. And then vast amounts of data. Wait a minute. Vast amounts of data is produced. You must be sifted and ordered for useful information to become apparent. So and then reference databases, SAGE reference databases. So everybody know about that. What is NCBI? National Center for Biotechnology Information, and then NLM represents National Library of Medicine, and then NIH represents National Institute of Health. GOV is the organization. See this result, the data results. So from tags to genes, collect the sequence record from GenBank and then assign the particular sequences. Extract 10 bases adjacent to 3' most of the sequences. Here see the sequences. Assign unigene identifier to each sequence with a SAGE tag. Okay, so see this web page, that is NCBI SAGE map V Northern. Here see this, that is a SAGE format, SAGE map, and then here see this SAGE map and then result NCBI differential gene expression by the SAGE, and then identification of differentially expressed genes in sample from different physiological or pathological conditions. So here the SAGE application of many statistical methods and then Bayesian methods, chi-square test methods. The three methods are playing the major role, and the SAGE software searches GenBank for matches to each tags that allows assignment to three categories of tags. And then first one is the mRNA derived from known genes, and the next one is the anonymous mRNA is also known as expressed sequence tags, and then mRNA derived from currently unidentified genes. And then here see this comparative studies of SAGE and microarray. What is mean by array? Array represents the consecutive set of memory location. And then what is the difference? Microarray and then SAGE. And then SAGE an open system which detects both known and unknown transcripts and genes. See this first example is the SAGE and then control RNA and then experimental RNA. Two samples are there. First we are generate the tags and the next step is the ligate tags, and the ligate tags என்ன பண்றோம் next sequences and database search. So and then final result is differential expression profile and then microarray. So see this control RNA and then control experimental RNA. So here it will present first step is the fluorescently labeled probe present, and then hybridize and scan the result, and then find the differential expression profile. That is a major difference between the SAGE and the microarray. See this comparison, the three major points, and then SAGE it will detects the 3' region of transcript, restriction site is determining factor, and then microarray is the target various region of the transcript base composition for specific of hybridization. And then SAGE collects the sequences information and copy numbers, and then microarray fluorescent signals and signal intensity, SAGE sequencing error and quantitation bias, and then microarray labeling bias and noise signals. Here see this difference. Detects the unknown transcript SAGEல பண்ண முடியும், and then microarrayல முடியாது. Okay, and then quantification absolute measure in SAGE and then relative measure in microarray, and then sensitivity compared to microarray SAGE is high and then microarray is moderate, and then specificity moderate in SAGE and then high in microarray. Reproducibility good for higher abundant transcripts, and then microarray have good for data from intra-platform comparison, and then direct test of the SAGE 5 to 10x higher than arrays, and then microarray have 5 to 10x lower than SAGE. So what are the recent SAGE applications? And the first one is the analysis of yeast transcriptome, and the next application gene expression profiles in normal and cancer cell, and then next application identification and classification of p53 regulated genes, and then analysis of human transcriptomes, serial microanalysis of renal transcriptomes, and then analysis of the particular genes, and then using the transcriptome to analyze the genome. That is a long SAGE. What are the limitations are available? That is an important one, and then quantitation of bias. What are the main advantages of SAGE? Generation of longer 3' cDNA from SAGE tags for gene identification, and then long SAGE and cap analysis of gene expression and then find the gene identification signature and then super sage and then digital karyotyping paired end tag and then what is mean by long sage increased the specificity of sage tag for transcript identification and sage tag mapping. And the next one is the collect the tags of 21 base pairs. Different type 2 restriction enzymes are used here and then adopts the sage principle to genomic DNA. Okay, see this diagram. And then capped CAG represents capped analysis of gene expression. See this diagram. And then micro sage, that is a one of the type sage type micro sage. See what is the difference between the sage and then micro sage. So மேஜர் ஒன் டிஃப்ரென்ஸ் அப்படின்னு சொல்லி பார்த்தீங்கன்னா இதில் சைக்கிள்ஸ் பிசிஆர் ஆம்ப்ளிஃபிகேஷன் சைக்கிள் சேஜ் வந்து 25 to 28 சைக்கிள்ஸ் அண்ட் தென் மைக்ரோ சேஜில் பார்த்தீங்கன்னா 28 சைக்கிள்ஸ் ஃபாலோடு பை RE-PCR on excited Titacs and then super sage. So here it will be increase the specificity of sage tags. See this, that is a super sage. And then gene identification signature. GIS represents gene identification signature and then paired end tag. So it is a sage, a look glass of for cancer. See, that is a cancer genome anatomy project related informations. And then P53 tumor suppressor gene, immunological studies, everything there. Okay, so what is sage? That's an important one. You study that topic, it's very useful for you.